Hello, my name is Graham Young, and I'm part of Team 4 with Hannah Barrett and Jeremy Earl, and we are using MATLAB to track cerebral atrophy and MRI scans for possible brain disease diagnosis. Our problem is, although many neurodegenerative diseases are able to be diagnosed with the help of imaging, doctors, and radiologists, once they are diagnosed, the symptoms are usually detrimental for the patient's life. These diseases are also very difficult to diagnose because of the symptoms of the multiple different diseases. They can be very similar to each other, which makes them difficult to differentiate between them. Diagnosing the correct disease is very important. As said before, many of the similar symptoms range across lots of neurodegenerative diseases. Creating a code to analyze brain shrinkage within each area of the brain would be the best way to do this because specific neurodegenerative diseases target different and specific areas of the brain. Now, why is early diagnosis for neurodegenerative diseases so important? Because time is critical. The symptoms can become more severe as time progresses. They are more likely to develop over time, and they can cause permanent damage to the nervous system. Earlier diagnosis can also mean earlier treatment, which can improve or control your symptoms, relieve pain, increase mobility, and maintain your quality of life. Our project aims to compare changes in the total brain volume loss shown through MRI scans to provide a possible diagnosis for neurodegenerative diseases. These MRI scans would be ideally taken from patients over extended periods of time and will be used to detect the changes in location and severity of brain shrinkage. Our code will be able to detect this brain shrinkage by thresholding images and calculating percent loss between the two MRI scans, and we'll be able to diagnose between certain diseases shown below. The data set that we used, it was from Kaggle, and the specific source is called MRI or Alzheimer's MRI preprocessed dataset. It is a dataset that provides 6,400 MRI images that have been broken down, ranging from demented to moderately demented, and they are 128 by 128 pixels in a JPEG file format. They are in a transverse. They show the transverse slices of the brain, and our dataset is not neurodegenerative disease specific and provides MRI scans of patients with undiagnosed diseases experiencing cerebral atrophy. First, it begins by comparing the two images using the comparison function, which blurs the two images and then provides a structural similarity estimate. During this comparison, the code employs color thresholding and image processing functions to create masks for different regions of interest. After a successful comparison, the code processes the images to identify specific brain regions and calculates the percent loss and misclassification loss. Subsequently, the analyze function divides the hue saturation value, or otherwise known as the HSV threshold, of a region into blocks, evaluating the percent loss in each block. Finally, the diagnose function then utilizes these percentages to diagnose potential diseases such as Alzheimer's, Parkinson's, ALS, frontotemporal dementia, and etc. shown below. So here is our code. Here we can see the three main functions: the comparison function here, the analyze section function here, and the diagnose function here. This mapping function here basically color maps the different percentage losses within each block and grades them to a certain hue or intensity of color. As we run the code, it will first ask us for a normal brain MRI scan. Then it will then ask us for a non-normal brain MRI scan. After running the code, we can then see the three main outputs below, being the total percentage loss, the misclassified percentage, and the anatomical structures with the most shrinkage. And using those anatomical structures, we can then do a suggested diagnosis shown here, which evidently is Alzheimer's, Parkinson's, or frontotemporal dementia. Here are the four main outputs of the comparison function. This right here is taking the normal MRI scan and the non-normal and stacking on top of each other. And it is measuring the differences in the pixels values, which can be seen with the green here, which is the non-normal compared to the normal, which would mean the brain loss. And the purple is the normal compared to the non-normal, which is the misclassified. The HSV threshold here is our brain loss, which would be in the green. Misclassified is in the purple. And our total brain area is the entirety of this image thresholded. Here are the results from the analyze section function. It takes the HSV threshold and divides into a 5x5 image matrix shown here. It also calculates the percentage of each of these tiny images here and has it as a percent loss from the total brain area. If you were to add up all the percentage losses of these tiny images, it would equal the total percent loss shown previously. And here's the color map with different colors shown below, showing the intensity of percentage loss within that 5x5 image matrix. Thank you for listening to our presentation, and have a good day.